excited for everyone to be here. Thank you so much. And really to kick things off is, among many things, he's the Executive Director of MGI, Dr. Jeffrey Milbrandt.
So for those of you who aren't immunized to subliminal messages, I hope you didn't pay too much attention to the earlier thing. Thanks, Larry, for the introduction. I'd like to welcome everybody to this event, Spatial and Single Cell Analysis Symposium, sponsored by 10X. This event is a reflection of MGI's role here at Washington University and in the St. Louis area. As a hub for personalized medicine, we try very hard to bring new technologies to bear so that you have access to them and you have the expertise available to you so that you can use them to answer the exciting questions that you find interesting. So our goal is to make these technologies available to a broad range of investigators with a broad range of expertise. Today you're going to hear from three members of MGI, Rich Head, Bob Fulton, and Wille Buchser, and they'll provide overviews of these new technologies. And I hope you see from their talks how the centers at MGI are designed to work together, from DNA, RNA sequencing to cellular imaging and then on to machine learning. So we also hear from WashU investigators Jennifer Fultz from the Division of Oncology, who works on NK cell biology and how it might be therapeutically applied for hematologic malignancies, and also then Sanjay Jain from the renal division. Now Sanjay trained in my lab many years ago, and I'm happy to report that he's recovered from that trauma and doing a great job now in understanding the pathophysiology of kidney disorders using single cell and spatial technologies. And then we'll also hear from our colleagues at 10X, who've been instrumental in making these technologies, making this event happen, but also constantly striving to make these technologies better. They are great partners for us at MGI and for many of the scientists who we both engage. And their dedication to raising the bar of these technologies from higher throughput, better resolution, and I understand, I hope this is true, reducing cost of these analyses, that they've really done a great job with that. So we thank them for supporting this event and being here. So I encourage everyone today to make today's event a true sharing of information. I hope you learn something from the experts that we've assembled. Perhaps more important, I hope you learn something from working with talking to each other. And to facilitate that, we're going to have a networking session after this is over at the end of the day, about 3 o'clock, where there'll be food and drinks, and you'll be able to engage each other on your problems or successes with single-cell technologies. So with that, I'm going to start the proceedings with Bob Fulton, who's going to go first. Right, Bob? Yep. Come on up.
All righty. Well, thanks all for coming. And I want to start out by echoing what Jeff said in terms of working with 10X and what great partners they've been through the years. I started my interaction with them many, many years ago at AGBT talking about long-linked DNA reads. So the technology has progressed significantly since then. So the agenda for my section of the talk here before I hand it off to Rich, we're going to talk about single-cell applications and sort of the standard 3', 5', the 3.1 and version 2, and then talk about the Flex and the GemX, which are single-cell technologies as well. A little bit of long read associated with those technologies, or some of those technologies at least, and then the spatial technologies, the Xenium and Visium HD. So single-cell gene expression, I'm going to breeze through these for the most part. These are pretty standard technologies that we have had for quite some time now, with 3' gene expression, various applications, labeling protein, surface proteins, for instance, CRISPR screening, and then mechanisms for multiplexing to squeeze out as much efficiency out of libraries that we can. Again, then 5' gene expression as well. And we exploit the 3', 5' depending on, regions of interest or things that you're looking for. So with the 5' gene expression, you get immune profiling capabilities, for instance. Again, surface proteins and multiplexing capabilities. And 20,000 cells for input into this process is a good rule of thumb. And then 3' gene expression and attack seek all from the same cells. So again, sort of expanding on the omics profile here, looking at open chromatin as well as gene expression. Okay, so the 10x Flex. So this is fairly new. This is a probe-based technology designed primarily for fixed samples, although you can take your fresh samples and fix them. In fixed samples, including FFPE. Again, it's a probe-based assay, and as you'll see in the next slide or two, it enables the probing of specific cell populations and then ligation and the combination of those, harvesting those ligated probes for subsequent analyses, but again, leveraging the ability to plex them. So these come in a lot of flavors. I had a discussion with 10x yesterday actually on how to convey this and how to understand these kits, where you can do this top, for instance, is 4 individual reactions. This is 10,000 cells per. You do the probing and you run it do the microfluidics to make the gel emulsions. But then when I talk about multiplexing, here there are 4 barcoded sets of probes that can be applied to different samples, combined, and then run through those same microfluidics, thus combining 4 different samples in a single run, so capturing some cost efficiencies there. This can be scaled up to 16 barcodes into a single reaction. That can then be combined, run through the microfluidics again, and assayed in a library. So you can see the impact of the cost. This is just the reagent costs. And there was a rumor flying around WashU. I got multiple emails about a $100 10x library. That came from the CEO of 10x. Thank you very much, CEO of 10x. But that's where they're striving to get over the next 3 to 4 years. So we do not have $100 libraries currently. But again, you see the sort of the benefit of this combinatorial mechanism through the gem creation and the sequencing. So the sequencing component of this is relatively minor in comparison to the library construction, and the sequencing costs continue to go down. So with this technology, one key is to count cells or nuclei accurately. That enables precise balancing of your pools. So we just recently upgraded our cell counting capabilities with this Nexcelome Celometer K2 cell and nuclei counter. So again, this gives us much more efficient combinatorial processes. Okay, so then another new technology is called GemX. It's just it's actually reverting back to an older way of doing things through the microfluidics that's more efficient, and I think there were patent issues and things like that had come up, but that's all been resolved, and we're back to this robust technology. So with this pivot, we are moving from the current version 3, 3 prime, and version 2, 5 prime, to version 4 and version 3. Confusing enough? So substantially more sensitivity, so up to 2 times more genes, and I'll show you a few graphs to demonstrate the sensitivity gains. It's built to scale, so it does twice as many cells, so we're targeting 20,000 cells versus 10,000 cells for the library. More cost effective, so again, twice as many per cell at least, more cost effective because of doubling of the cells, and there's a minor reduction in the cost of the kit as well. Enhanced quality, so 2-fold reduction in multiplet rates. Maximum sample recovery, so typically that number's around 50 to 60% of cell recovery into the box and out of the box as single cells. That's upwards of 80% with some cell types now, so that's improved. And then again, just improved robustness in terms of redesigned microfluidics, so less clogging and things like that as well. And then anecdotally at least, longer full-length cDNA, which will come into play as I talk about the long-read technology. So here's a few graphs. So the lighter blue color here is the 3 prime and 5 prime current iterations, and then the GemX is the darker color. So these graphs are showing median genes per cell. And then this is just the number of reads per cell that can be applied. So one interesting aspect, so see how these are flattening out. So we've really, at 50 to 60,000 reads per cell, we've really depleted the uniqueness of genes in the library, where the trajectory of these with deeper sequencing, it is there. So that actually, we can apply other technologies as well, for instance, hybridization. If you have genes of interest that are lowly expressed in the background of higher expressed genes, we can pull things out. So I am very encouraged by this. And then, again, 3 prime gene expression. So this is the number of UMIs, or UMI counts per cell. And again, you can see significantly more unique molecules per cell in these libraries for the sequencing. Okay, so I mentioned long-read technology. So out of the box, we get full-length cDNA, or full-length cDNA. It's a relative term, but reasonably long transcripts, which we can capture and sequence on long-read technologies. Now, for these sorts of assays, they're counting assays to some degree with unique molecular indices, multiple genes, multiple cells. So depth is a consideration. From these long-read technologies, you're going to get less than 20% of like standard reads compared to short reads. But you're getting full-length transcript. So for expressed variants, splice variants, things like that, you're gonna, you can see those. And we've worked on some enrichment strategies, both hybridization as well as a CRISPR depletion to deplete commonly or highly expressed things. So we're applying both the PacBio and the ONT for these applications. Okay, so spatial. Of all the things I've seen in my career, this is one of the most amazing, to be honest, to be able to see transcripts in the cell. It just amazes me. And so this is the 10x Xenium. We have a couple in our lab. I'll talk more about those. This is the CytAssist, which we use to transfer material between, for instance, a slide from the Xenium onto a Visium or Visium HD, as well as from older slides to transfer material again to the Visium. All right, so 10x Genomics, or the Xenium, we installed this in May of 2023, so we're sitting on almost a year now. And I put a few numbers in red just to show you the progression over the last year. This is a ever-moving technology. So again, fresh or FFPE workflows are possible here. When we started, we were 350 genes for custom panels. We're now up to 480 genes and you'll see soon that that's a moving target as well. You can do custom spike-ins of 50 or 100, within 50 or 100 gene increments on top of those. So if you have some assays that you want to apply other gene sets to, or you want to add additional content to your probe sets. Pre-designed panels also, I think the biggest pre-designed panel I'm aware of is 380 genes at the moment. But again, you can spike in 50 to 100 additional genes. It was a 72-hour run, is now a 48-hour run on the instrument. And it's 2 slides per instrument run, and it's a 12 millimeter by 24 millimeter space, so a relatively large space, and the imaging time is about the same depending on the coverage there. But if you have multiple tissue sections, as long as they're not overlapping or sitting on fiducials, you can put multiple sections on a slide. The tissue is preserved throughout the process. Sorry, I skipped the proteins. So proteins are not yet available, but come in, I think, Q2. I'll talk about that in a minute. The tissue is preserved, so you can, after doing your xenium run, you can do H&E staining, immunofluorescence, other staining, and other applications, including the transfer using the site assist to the Visium for FFPE, or likely the Visium HD. I think that's not been vetted yet, but will be. And then you can couple all of these technologies, so you can do single cell assays and couple those data and integrate those data with the spatial data. Current panels, so this is the current offerings from 10X, like human and mouse primarily, or exclusively actually, with the most recent, so this is a slide again from this, I think this is from August when Rich and I did a talk. And we've updated some of the numbers. There's 282 genes on the skin. This is a brand new panel that has recently come out. But the skin and the colon weren't available yet at that time. So again, continuous progress. So this is just showing the engagement with WashU. So since we installed our xeniums in 2023, we've designed over 4,300 custom gene targets. And we've been on the cutting edge, which is the next slide, basically, of advanced customization. So designing for isoforms, single nucleotide variants, reporter genes, and advanced species and more. So we've got 11 committed labs to Xenium custom panels, 144 custom slides committed to design. So a lot going on, and then countless more conversations. So advanced customization enables broad applications. So this is all at WashU. We've done isoform / translocation designs, diverse species, xenographs, exogenous sequences, so viral sequences or reporter genes, for instance, express single nucleotide variants, and then TCR, BCR profiling is relatively new. We haven't done that yet, but there are capabilities. Okay, so what's coming next? And coming or has come. So multimodal segmentation is here. We have kits. I think you may see data today from the first run with the multimodal segmentation. 5,000 gene panel, and I'll talk about all of these in a bit more detail. Inline multiplexing of proteins, again coming second half of 2024, and then 1,000 to 2,000 gene plex panels. So I've had multiple conversations with 10X. I sat through a workshop in February where we had just open discussion on what the right strategy is and what the community wants. So your voice, please be heard. Please engage with 10X. They're interested in people's perspective. With more probes comes longer run times and more expense, but more comprehensiveness versus shorter sets. And they provide some flexibility, but again, they're they're interested in engaging to set this direction. Okay, so multimodal morphology-based cell segmentation. So this is a cell staining reagent. It's currently, you can apply it for a fee, but there are boundary proteins, interior RNA staining, nuclear as well as interior proteins, and then algorithmic development to better do cell segmentation. So just to show you sort of the outcome of that. So this is prior to the cell segmentation reagent. So these are macrophages actually, which you can see. So you can see the difference between this image, which was the original, versus applying the cell segmentation reagent to better cell segment. And then here's just a few more examples utilizing this reagent and the cell identification from these various tissues. Okay, Xenium 5000 plex, so this is 5,000 genes. They do have gene lists available, so if you're interested, reach out to me and we can get that information to you. Improved chemistry allows for this higher plexing. Their goal is to maintain the sensitivity, so down to single transcript sort of levels, while still being able to look at 5,000. Dilution of highly expressed genes so that you don't swamp out signal, but then calculating sort of relative levels from that. Practical to run in a working week. So roughly, so if it's 5 to 6 days. I've heard both numbers. This is the most recent number I said, or I've heard, so we'll go with 6 days for the slides. Just to show sort of sensitivity. So this is per cell, transcript per cell. Pretty amazing sensitivity. Looking at 5,000 genes across different tissues, and of course, different gene expression for the different tissues. Proteins, so this is coming latter half of the year. And again, on the same tissue section, the idea is that they're going to have about 40 to 50 protein conjugated antibodies for looking at proteins at launch, or that's the aim at least, with the idea of applying 20 for a gene set. Again, on the same tissue at the same time. And this is just a picture of a 20-plex protein and a 1,000-plex panel, so cutting edge on both of these. But again, you can see the proteins and the nucleic acid. And then Visium HD. So this is stepping away from the Xenium. This is a standalone, this is a slide basically with two sections, two 6.5 millimeter square sections on the slide. It's compatible with fixed or frozen. It utilizes the site assist to transfer nucleic acid to the slide. It's feasible to transfer Xenium transcripts to the Visium. So we have not done this at this point, but we do have, we have the equipment and the know-how to do this, where you could transfer from a Xenium slide with the granularity of your gene panels to the full transcriptome. Now there's a different size, but you can outline your 6.5 millimeter square region, pull this down and do a full transcriptome at nearly or single cell level. So the way that this slide is designed, so for those of you who have done Visium or Visium for FFPE, those are 55 micron circles and they have gaps between the circles. This design is a contiguous lawn of oligos, and they're in 2 micrometer square regions. The analysis packages are built on 8 micrometer sections, but you can drill down all the way to 2 microns. on these. And again, whoops. There we go. Again, 6.5 millimeter surface area times 2. So we're working, so again, this is sort of bleeding edge. So we're working with 10X who, so we want to validate this on the HiSeq X Plus. So it takes about 300 million clusters to sequence this, the library from this. So we're working with them to sequence these on the new sequencing platform to grab the cost economy of all of this plus the sequencing. And again, there's just subtle hitches always with these new technologies and trying to figure these things out. So we need to develop this and work closely with them. So again, I want to thank Spence and Connor, Amanda, Larry, for a lot of this content actually, because again, we don't have it yet. But it's quickly coming, and it's very nice to be engaged early on to then provide that to you. So if you're interested in any of this, I'll be around all afternoon and happy to discuss. And Spence, I know, is going to fill in some of the technology details, but I'll hand this off now to Rich, who's going to give you more of an analysis look and practical application.
Yeah, I'm your dose of reality for the morning. Okay, so wonderful new technology, right? But there are some practical realities to a lot of this. And that's kind of my job, is to not necessarily be the wet blanket, but kind of let folks know that if you want to do some of this stuff, you really need to come talk to us, especially with Spatial. The days of, hey, I've got a bunch of samples, my lab tech's going to come over and drop them off in your freezer, let us know when the data's ready. That is not how these spatial platforms work. That's not how the interaction goes anymore. It's a lot more involved, a lot more complicated. It works, and we've got a lot of good partners that work with us to get this done and get you back good data, but you really need to work with us on these for two reasons. One, it is a complex process, and two, as I'm sure you're aware, it's not cheap. So when you're doing this, you want it to work, and we want to make sure that it works for you. So not so much in the single cell space. I think a lot of folks are familiar with that process, and I'll speak briefly about single cell and some of the new technologies that Bob covered. But when we get into the spatial, I really want to stress that this is a process. We really need to work with you to get you to a successful conclusion on it. Okay, so this is just kind of an overview of the roadmap that Bob and even the previous slides from 10X had mentioned. We've launched much of this. There's just a couple of exceptions. As Bob mentioned, the xenium proteins aren't quite ready yet. We're just in the launching phase. It's not quite ready yet. We still got a little bit of work to do here, but it's very, very close. We do need to do some validation on the NovaSeq X. We get to be a little bit of a guinea pig on that. But we want to make sure that it's working properly before we release it out to everybody for use. Okay, quickly through the single cell, because again, a lot of this has been covered, but just kind of bring everybody back to some of just some key points. When you want to do a single cell experiment, again, if you've not done them before, these require advanced scheduling with our laboratory. So Jen Ponce's lab will work with you to schedule delivery, because most of these require viable live cells and or nuclei in suspension. And so as a result, we need to schedule it so that you bring over your cells and they're still in their somewhat happy state in order to do the experimentation. There are a number of things that Bob had already talked about that, kind of the older technologies that we had, the regular 3' version 3.1, version 2, which is what we've been running. We're moving up to the new features that are the new protocols, basically, that do have a number of advantages. Basically, the recoveries, the enhanced transcript recoveries you saw is substantial compared to the prior versions for single cell. So this is something we're very interested in seeing. Couple notes, some of these things are CRISPR compatible, some are not, as well as I believe that the prime is, from what I understand, the version 4 is not CellPlex compatible either. But other than that, these are fairly substantial advances on the single cell front. Bioinformatics. This is the big question we always get. Okay, fine, I'm dropping stuff off. What am I getting back? And this is an issue for a lot of laboratories that we work with. It's somewhat in the single cell space. There are a lot of tools out there now in the single cell arena. We do run some standard pipelines and workflows in GTAC where you will get, you're not just going to get raw data back from us. There are some things that you will get back from us beyond that use the Cell Ranger platform as well as Seurat 5 for some of the different workflows that we run. We can also, if we are given the information at the time of submission, our bioinformatics team, and there's sufficient replication, et cetera, can also provide information in terms of differential expression from some of the original protocols. As you imagine, the stuff that we have built for the original 3', 5' fixed kits is going to transfer over to the new versions. So all of the stuff that we're doing today and can provide as output will transfer over. For example, the Cell Ranger output, you get HTML files with alignments, barcoding, feature counts, et cetera, TS&E plots. In addition to that, we do have the Seurat 5, which provides quite a bit more information that we supply back with the knowledge, as well as the Velocity of the Loom workflow, where we can also, as I said, if you give us the information ahead of time, we can provide information about differential expression. So again, single cell's been around for a while. It's changing, evolving, getting better. But this is, I think, fairly well known by a lot of folks in the community that use the technologies. Now, spatial. I can't even put this because there's so much that I have to sort of communicate here. I couldn't put this all in one slide. So basically, I'm going to go through each of these as a set of slides. I apologize for the wordiness. I'm going to try to pick out the highlights here. Another thing that we've talked about is we want to try to get all of this incorporated into our website. Jen said she gets lots and lots of questions about this stuff all the time as we're doing these experiments for folks, and we want to try to make sure we're getting this information to you ahead of time so that when you're preparing to do one of these experiments, you at least are sort of forewarned as to what the process is going to look like. So the original Visium and Cytosys workflows, as Bob had mentioned, this is basically a 50 micron resolution. These are 50 micron resolution spots. This is sort of a whole transcriptome type of workflow. This has been out. This is the oldest of the spatial technologies that we've been working with here. This is, oops, sorry. I am having problems with this. Going all the way back. Trying to hit the pointer. Okay. FFE blocks or pre-section slides can be accepted. H&E staining and 20x images can be done on the same slide. We work with the DDRCC, and I'm going to talk about this a great bit more in the next with the Xenium. And the Wookiee can help with this. And effectively, these images can be sent to in TIFF file format to GTAC for final analysis. I'm going to walk through what kind of these workflows look like in a graphical format in just a minute. So you can kind of get a better feel for what this entails. One thing that I will point out, fresh frozen is available, but from what Jen said, we've not done this. So it is an available technology. We've not done fresh frozen on the older Xenium platform. Okay, sorry, excuse me, Visium platform. Xenium. Panel-based, as Bob mentioned, and these can be custom designed as well as the 10x off-the-shelf panels. We do accept FFPE blocks, and I'm going to show you in a second because that involves a bit more of a process. So here's where things start to get complicated. If you are very comfortable in your laboratory making these slides, by all means you can do that and you can deliver the slides to us. It's a very involved process. You need to get your images, you need to get these sections aligned properly on the slides, et cetera. There's quite a bit to it. If you are not comfortable doing that, and I'm going to show you in a second, we have partnered with the DDRCC. This is a very, very knowledgeable, experienced lab in this space. We work with them. There is a back and forth process, and we're trying to streamline that because for the investigator or the laboratory technicians for the investigator, there's a good bit of back and forth that goes on with the samples before they finally get to us to run on the Xenium to make sure you have everything you need for a good, successful conclusion at the end. So both, as I mentioned, both FFP blocks and fresh frozen slides are accepted. Okay, so here's a little bit of the overview. I'm going to step out a little bit to kind of get to this a little bit better. So first thing you need to understand is what panel you're going to use. Are you going to use a custom panel, or are you going to use one of their off-the-shelf panels? That's sort of point number one. Once you have made that decision and you have your blocks or your sections you want to work with, now you have to decide what you're going to deliver. Are you going to deliver slides to GTAC, ready to go, or would you prefer to work with the DDRCC to section your blocks and create the slides that will come to us? They're very, very good at doing this. This is a very knowledgeable lab. We've been working with them from the beginning on this, so we've partnered with them for some time. Deb Rubin's the director. Tai-Cheng Liu is the co-director of the laboratory. So we have a formal arrangement with them to be able to do this. In addition, there's another piece to this, other than just the DDRCC sectioning. Again, we're trying to figure out how to streamline this, and that is the images. Often people are getting done at the Wookiee. So there's a bit of back and forth that's going on here. There can be a delivery of the blocks to the DDRCC, then taking those in order to get the images done at the Wookiee, and then bringing things to the GTAC. There's also a step of doing an RNA quality control test. So we need to make sure before we run these that the RNA is sufficient quality. That was also requiring a transfer of material back and forth, but we're now working with the DDRCC so that hopefully they'll be able to do that process there. So that will eliminate one back and forth with these samples before the slides eventually come to us to run on the Xenium platform. So I'm not trying to scare anybody away here. I'm just letting you know we do know how to do this, but it is a bit of a process. We will work through you. We'll walk you through the process. We know, we'll work with you. The folks at the DDRCC will work with you, and we can show you how to get this done to a successful conclusion. But just pointing out that there are some complexities to it. This, like I said, it's not just, here I'm going to drop off some samples, let me know when my data are ready. Okay, as Bob mentioned, we're still like in the testing processes with this. It's here, it exists. It's not a to be announced or to arrive. This is something we're actively working on. We're actively testing the Visium Site Assist HD. I won't go through a ton of this because Bob's already covered a lot of this. The thing to remember here that's a little bit different now with Visium from Xenium. Xenium, you get your data directly from the Xenium instruments and our processing of that information off of the instrument. With Visium and Visium HD, these samples then go on to be sequenced. So it eventually becomes like an RNA-seq experiment at some point, where once you basically have your libraries, those then go on to get sequenced. So there's an additional step. Because of the density of Visium HD, the amount of sequencing that you potentially may need to do with these is probably more than you would be doing with the standard Visium, okay? In order, especially if depending on how much of the slide you've covered. Because as Bob mentioned, it's a grid, it's a full grid. And so if you've covered that full slide and you basically want any depth at all on those little 2-micron grids, you're gonna have to sequence to get it. So while the original minimum 275 is given, we're actually being told probably you want to double that or go up to at least 500 million reads for these if it's a fully covered slide. If it's a partially covered slide, if the section on the slide is partially covered, then you may not have to sequence as much. But if it's fully covered, obviously you're going to want to sequence more depending on how deep you want to sample into the transcriptome. Okay, bioinformatics for spatial, because a lot of this is very, very new. As Bob mentioned, some of these technologies just started to hit us last year. And the Visium HD is brand new. We really aren't even running that yet, and so we're still putting pipelines together. Visium and Visium HD will work with Space Ranger and Seurat 5. So we already have work pipelines set up for Seurat 5. Eric's group, as we're using, he actually has some data that was given to us by 10X. They're working to have Seurat 5 working with Visium HD as soon as we have Visium HD ready to go in the laboratory. So we're hopeful that that workflow, Space Ranger plus Seurat 5, will be ready for you as soon as we have Visium HD ready to go. And the Xenium there's a bit more to that. So there's the onboard analysis, which delivers some certain components. And then we have added not only Syrad 5, but SquidPy, which generates a lot of additional QC information about how some of the different transcripts break down on the per cell basis, et cetera, et cetera. So the Syrad 5 plus SquidPy workflows are in place now for Xenium, and we can deliver those data as part of the Xenium workflow. Okay, so now what happens if what you get back is not all of what you needed? You want to learn a lot more than just what you're getting back from these standard workflows and pipelines. There are some additional resources. Now, what you will get back from GTAC, you will get back, if you do the experiment with us, you will get those data back that I just mentioned from those pipelines and workflows for those different protocols. If you're looking for more assistance, looking for more help, and we get these kind of requests all the time. So while we have a very substantial and very experienced bioinformatics team in GTAC and in the MGI, it's still a finite resource. So the amount of collaboration they can do, or the amount of above and beyond that work they can do with investigators that may not have necessarily bioinformatics support in their laboratory, or not a lot of bioinformatics support, there are some potential other resources, and I say potential because you may or may not be able to identify collaborators. So there is a bioinformatics and computational biology resource portal here at Washington University. It's only visible to the folks at the institution behind our firewall. We have 20 contributors, expert contributors on the site now. It's growing. In fact, I'll put a plug out. If we've got any experts with spatial and single cell in this room in bioinformatics, we're always looking for potentially more contributors. These are folks that are potentially willing to collaborate with you on bigger projects and interesting projects that you might have. Basically, what you can do is you go into the site, you put in it takes literally 2 minutes to fill out the little form that says, I've got single cell or spatial data, I'm looking to do a publication, I really need some expert bioinformatics help to do this. Is there anybody out there who'd be willing to collaborate on this? It will go to the 20 contributors on the site. They will see it. If one of them is potentially able or interested in collaborating, they would contact you about the collaboration. And we have had over 30, this just went online about a little over a year ago, and we've already had 30 successful collaborations on the site across a multitude of needs in bioinformatics and computational biology. However, I will tell you, even this is a finite resource. Especially lately, we've had more requests than the contributors have been able to pick up. So some requests have gone unanswered. But if you need help and that you don't have, you can't find it, this is a potential site where you can get some help. So one other thing that I will mention is there are expert laboratories, not only in single cell, but spatial already. It's kind of a follow through from that last slide. So Ben Humphrey's lab has developed some wonderful bioinformatic tools that can be used with some of these platforms. They've been published. They're freely available. The downloads are associated with the publications, or Ben's lab can probably tell you where to go get them. They're in addition to the workflows that we've built, and they do some of the things that our tools don't do. So I just want to give kind of a shout out to them because they have built tools that are usable by others within the WashU community that are fully published and can be downloaded and used freely. So it's kind of a summary of everything we've been talking about. As you've seen, the Untitled Genome Institute has made a very substantial investment in spatial and single cell, and we have focused on the 10x platform. What Bob didn't talk too much about here today because there was just so much technology to discuss is a year and a half back, we did a very, very thorough analysis of all of the platforms in this space. We looked at everything. We looked at NanoString, VisGen, Akoya, which is more protein-based, et cetera. And in fact, I think in the talk we did back in the fall, we talked about it, that evaluation that we did. And just based on the roadmaps, et cetera, no one technology was perfect at everything. But what we saw with the 10x platforms, and this is not a commercial for 10x, this is kind of a more commercial we did our homework on this one, and we thought that 10x was by far the most comprehensive and integrated of all the workflows where we could give the best and most extensive offerings to drive research forward. So Even though they're the sponsors today, I would have said that even if they weren't the sponsors today. Okay. Xenium's online. The original Visium Site Assist, they've been online for a while, that Bob mentioned. Site Assist HD, Visium/Site Assist HD, online soon. We don't have the, basically you'll just be seeing an announcement come out when we're ready to go with it. As you know, GTAC's not shy about sending out emails and announcements to let you know when new technology is online and available, be on our website, et cetera. So we'll make sure everyone's aware of that. And in terms of the bioinformatics, we're continually, what I showed is what we have today. Eric's team is working hard to basically continue to build upon those workflows to generate more and more data, useful data, data for analytics and interpretation of these types of technologies as we speak. It's amazing what they've been able to do in a short period of time, and they're continuing to go. And one of the big things, is Bob mentioned, with the integration of things like spatial and single cell, Eric's team also has this on their roadmap to build a number of tools into their workflow to be able to integrate single cell and spatial. Eventually, even, we want to try to tie these things into bulk RNA-seq. So that is on the bioinformatics team roadmap. I would say I think we have a little bit of time now, so Bob and I can probably answer questions that anyone might have. One more thing, the cell segmentation. So I will tell you that Eric Tixon, who's our lead bioinformatician on this, has looked at the new data coming with the new multimodal segmentation and said it's phenomenal. He said that it's just light years better than doing it with just the DAPI staining. Now I will also throw on the bit of reality, it also adds some more expense, okay? So it is much, much better, but it does add some expense. So you will probably have to evaluate whether you want to add that expense or not, depending on the types of cells and tissues you're working with and how difficult the cell segmentation has been for you based on that tissue type. Using just the DAPI staining, we're really just seeing the nucleus, and then it has to kind of guess, right? If you're using like cuboidal epithelial cells, probably not such a big deal. If you're working with neurons or macrophages, probably a very big deal, and that's where the multimodal segmentation might be worth the extra expense. Okay? All right, so I'll stop there.
I'm Wille Buchser, so I'm right, the fourth floor is where all these amazing instruments are and all the work that these, Rich and Bob are doing. If you come down to the third floor, that's where genome engineering is, where Xiao Xiao is building cell lines and animals for people. And right next to her is my group, Functional Imaging for Variant Elucidation. And so as soon as spatial came out, I was like, oh my gosh, this is imaging. We have to jump on this and really think a lot about it. And I hope to think with you guys, just to think about, even if you throw out every single transcript and just look at the images that you get off this, you have an incredible data set, right? So do not for a single second think that, you should just instantly go to a UMAP and forget about the spatial. Like, that's why you did it. But the spatial itself is an amazing, amazing data set. And hopefully we can help you guys think of different ways to approach it and to really suck all the data you can out of it. And so on the far left, I have Lena, who is one of the research associates in the lab. And she's the one who's kind of pioneered a lot of this analysis that you're going to see. The samples that I'm going to show it on is mostly 10x Xenium samples that have gone through the instruments on the fourth floor. And Aldrin Yim is the one that's been kind of dealing with that and pioneering those sample preparations. And Nick and many others in the lab have been helping to build these up as well. And of course, Jeff Milbrandt, Rob Mitra, Rob Giroux are responsible for these tissues and these projects. So I'm going to tell you just for a second about what we do in general before we zoom into the spatial things and think about how that could work. Okay, so again, our goal is functional genomics, right? We want to answer questions about what is the cause, what happens when you get a variant, a mutation in a cell, what happens functionally, how does that cell change? And so our usual way to do this is kind of like CRISPR screening, kind of like FACSeq, but it's to do pooled image-based screening. So this is quite a bit harder, but it's something that the new spatial technologies are also going to enable, not necessarily today, but it's coming very quickly. So the way we do it right now is we usually are looking at cell types of interest, like neurons, and we're asking what happens if you put this genetic mutation in a neuron, but we're not doing that as one-off, we're doing it at a scale. So we're asking about thousands of mutations all at once. We have a system called RAFseq where we plate the cells with the mutations out on these releasable microrafs. Then we can do high content imaging. We can look at these single cells in really high detail, pick ones out that have phenotypes that you or we are interested in. Usually the cell, either something that makes the cell look pathogenic like a pathogenic mutant would, or something that corrects a pathogenic phenotype and brings the cell back to wild type and rescues it. So we can physically pick those out with an automated system and then go in and do single cell genotyping to figure out what was the guide RNA. We can do single cell RNA, whatever we want. So that's kind of the standard, that's our DNA. That's what we usually think of. And we've really pioneered this in the situation of neurons. So here you can see single iPS-derived human motor neurons. These little numbers that are hard to read, these are the ID of the raft that all these are sitting on, where we can go in and capture them. But of course, since these are single cells and these are confocal images, we can see in really, really high detail any organelles that we want to look at. Anything subcellular is really easy for us to look at and featureize and analyze. And I'm showing a few different cell types here. And even that space, like just looking at a single cell and analyzing the image data for it is an amazing data set, right? So now when you have spatial and you can do this across tissues where all the cells have meaning is just a glorious thing. And we have, I'm going to skip here. Okay, so anyway, that's where we're coming at this from, is we want to be able to pull as much as we can from the images. So each of these slices, just the image that you guys are going to get, these are data sets, right? And you want to get whatever you can out of these data sets and hopefully connect it also with the transcript level data. Most of what we're doing is with Xenium, but with things like the cytosist, you could also imagine growing your transcript space quite a lot. And so we can think of this at different scales, right? So of course, in a human brain slice, this is a mouse brain slice, you can look at different anatomical regions. You can zoom in and look at cell-cell interactions. You can look at, and then I see things like cell structural relationships. For example, let's say you're looking at cartilage and you want to know about extracellular matrix and how it interacts with the cell. Now that since everything is still in context, you can ask all those questions. And I'm using Prezi, so I have to take advantage of it. This is real DAPI-stained Xenium data. If you've only got images of DAPI, you can still ask incredible amounts of questions. What we're going to hear about is you'll have the option to have other stains in there as well. And obviously, the more things you can stain for, the more questions you can ask. We love to stain for organelles. We love to ask questions about mitochondria and Golgi, because for many neurodegenerative diseases, they are impacted at the single cell level when patients have variants. But even here, you can see at the resolution that these images were taken of this mouse brain, every individual nuclei has different, it's staining, right? How the DNA is stained, what do the nucleoli look like, what are the heterochrome and euchrome. You can see a lot, right? So just, again, even at this single cell level, there is a lot to be learned. And don't throw it out, right? The reason you have this image isn't just to segment and ask where the transcripts are, right? So the challenge for this whole talk is think top down as well. What can I get from the image? And then I'll ask questions about the transcript based on differences in the image. So even here, you guys can come up with hypotheses about different nuclear subtypes, some of which you can see are very bright, some are dimmer, some are smaller, bigger, right? So let's say, how are all the dim nuclei, what are their transcripts, how do they differentiate express from really bright nuclei, right? Very relatively easy to do, but that's one thing you can do. Also, anything about cell-cell interactions, these cells that are in this really dense granular zone versus the cells that are distributed outside of the granular zone, lots of questions there. Now we can go zoom out a little bit, look at a tissue level. You can see the dentate gyrus here and the nuclei in the dentate gyrus. You can see other really interesting microstructures like these lines, right? So this is probably where vasculature is moving through the brain at that level. And so those are the cells that are right alongside of the vessels. And a common thing that many of you guys who are used to looking at this data is, well, sure, if I have Visium level data or Visium HD level data one day, I'll be able to say, here's all the possible transcripts that these perineural cells, these near blood vessel cells are going to express. Maybe I can try to do it bottom up. I can say, oh yeah, those are what those express, so I should be able to identify those. Right now, do you really want to waste your 350 gene xenium panel on markers for cells like this, when the image tells you exactly that that's what those are. So many things you can take from here. We can zoom out a little further and ask, now we can see regions of the brain. This is one of the hemispheres, right? What are all the different layers of the cortex? What are all the different layers inside the hippocampus? Wouldn't it be nice to just have those annotated, and now I can ask questions about what's layer 3 versus layer 4, top down from the image down to the transcripts. Or maybe I want to do cytology. Maybe I just want to know, in these samples, I have a control and a disease. What is the count in a particular layer? How do those cells look different? So hopefully I get you thinking more about this. So the subcellular features that you can measure, even here with just nucleus, there's a lot you can measure. You can do cell type classification really well, and I'll show you a story about this. Again, even just with DAPI to say these are different cell types that we can identify top down and then apply questions about transcripts to them. We can look at clusters of cell. We can look at lammas, the contours, the stria. And of course, you want to eventually get very anatomically precise about labeling these areas. And I've been using the word top down. What I mean there is like image to transcript, right? Ask a question about the image, like how does brain region 1 differ than brain region 2? Now we can ask about the transcript. And of course, we can go bottom up. We can say, based on a transcript to define a cell, now let's ask how that region in the tissue looks different, right? Is it bigger? Is it shifted? Is it smaller? But do be careful about double dipping. You don't want to do both. You don't want to define something with an image and with a transcript and then ask about a transcript or ask about the image, right? Use one, ask about the other is the safest. And please stop me at any time. You don't have to wait till the end. But so far so good. Anything you guys want to chat about? So a lot of, up until now, we've been talking about this word segmentation, right? So segmentation is important because if you want to turn your data set into a UMAP, you need to know what transcripts belong with which cell. And segmentation is the process that says like, oh yeah, here's the region that defines cell X. All the transcripts we find inside of cell X, we're going to assign them just as if it was a single cell gem. Those are all with the cell, and now we know the XY coordinates of it. Great, and now here's another segmentation region that defines cell Y, and we put those transcripts together. But in addition to answering that question, where do the transcripts go? And I think what Spence is going to tell you in detail about is if you can not just know the nuclear boundary, but of course know the whole cellular boundary, now you can take transcripts that were kind of between nuclei and do a better job of assigning them to nuclei A or nuclei B, or cell A or cell B. But again, you might not want segmentation just to know where your transcripts are. You might want the segmentation to ask questions about the biology, which is what I think we all really care about. So think of segmentation more broadly as just a spot to throw transcripts in. Okay, I'm going to show you a few vignettes. The first is with human sciatic nerve tissue. The second was mouse brain, and I'll show you a little bit about spinal cord and some other techniques. Okay. So human sciatic nerve. This is actually where we got our start with working with this xenium data. And again, it was just like so fantastic to have this data set. So this is through Rob Giroux. This is an actual human sciatic nerve, so from the leg. And you're seeing a cross-section, so the axons would be running towards you or from the screen towards you and back again. And even at this view, you can see the fascicles, which are those big clusters that contain lots and lots of axons. And of course, within these bundles, there's also Schwann cells. So there's both axons, Schwann cells, and a few other cell types. And so, let me see if when we zoomed into this, zoomed in really far, it was really, really hard to segment with the standard software that comes with the Xenium. It was really hard to segment with CellPose. It was really hard to segment with Baser. And you can kind of see why, because the axons, which of course don't have DNA in them or have very, very little DNA, they show up as a little, like a ghost, a ghostly shadow. And then the probably Schwann cell nuclei show up, of course, very distinctly and very brightly. And as you can kind of see from this, sorry, I should have grabbed a higher resolution image for you, but there are thousands and thousands of axons inside of each of those fascicles. And even within a fascicle, the variation of intensity of the nuclei and the axons was really extreme. So any kind of usual, again, this is why most things were failing, is because there was just all this background of the axons and very broad intensities of the nuclei. And so several methods were tried and they kind of all failed. And so this is where we use some of our techniques that we use all the time with cells came into play. And this is what Lena kind of pioneered. And so this, which is now really hard to see, but I'll zoom in on it in a second. So what we did is we had what are called physics or biology-based machine learning algorithms that can go in and look for these structures, whatever structure you guys want, whatever structure you care about, right? Whether it's immune cells and macrophages, whether it's cancer cells, whether it's different types of neurons like DRGs that are just different than other cells. And so we had an existing pipeline for this where it happened to not have anything to do with axons and nuclei, but it was the same idea where you had two different classes of structures. And all of our stuff, since it's physics-based, we can just run a few more simulations and then retrain, and now everything is like perfectly tuned to the task at hand. And so what you can see if I zoom in here is that now this algorithm, which was pixel-based, every single pixel was defined as either background as black, blue as an axon, red as a Schwann cell nuclei, and it even got some of the edges as this like perineuron. And so already you went from this really, really hard to segment thing to something that was really easy to understand what was going on. And we and the other investigators got really excited about that. This is the pixel-based segmentation, or really classification, right? So every pixel is classified. You actually get borders of the nuclei themselves. You can do various instance-based algorithms to progress this forward. But of course, as soon as you do something, you got to benchmark it. So generating all the data and generating those fancier segmentations, none of that is done with hand labeling. So no human had to go in and hand label pictures to train. This was done with deep learning, of course. But all of the way that it works, everything is generated with simulations. Nothing is hand labeled. But to benchmark it, that's when we had to go and actually do some hand labeling. But just so you know, so if you see anything with hand labeling in my talk, it is never used in training. And that's also really important because it means we're always inferring out of context. And so when it works, it works really nicely. And so a bunch of hand annotations was done on the different types, whether it was axons or nuclei. A little bit of fun here just about how you compare and contrast, right? Cell pose, Buchser, the canned algorithms all have different kinds of outputs, whether they're based on pixels, whether they're based on polygons, whether they're based on masks. And so we did several things to try to cross-compare all of these and really not bias ourselves towards one because they just gave us slightly different output. And what we found was that our ways of doing this were like insanely better than cell pose or Buchser or many other alternative approaches. So the ability to look into this tissue, not only segment all the nuclei and the axons, but also get really good classifications between which were which now we can overlay transcript data onto, was worlds apart from what was available at the time. Now, CellPost 3 has just come out. I think that that's going to do better, but it's still going to need a lot more special hand labeling to get it up to par. And we were talking about if we had other stains, we could very accurately say where the edge of the cell is. But this is how the edge of the cell was called previously, and this is after the new algorithms trying to call, how to sort the transcripts and which buckets to put them in. And so one of the fun things we can do with this now is say, again, ask questions from the image down to the transcripts. And so one of the questions that we asked, if you guys know about these peripheral nerves, is that generally large diameter axons, these they're myelinated and they have certain transcripts associated with their Schwann cells. Small diameter axons occur in bundles and they're not myelinated the same way. They don't have the same kind of Schwann cells. And so we just asked, what are the genes that are most linearly correlated? We did linear regression amongst all the transcript counts. And what we saw is that for the large diameter axons, which are over here, the top 2 genes are exactly what you would expect. In fact, the top several. So all of these are myelin-associated genes that are expressed by myelinating Schwann cells. On the other side, which is the red on the top, for the smallest diameter axon, these are genes that are known to be present in these special Schwann cells that myelinate small diameter axons, right? So very quickly we could pull biology from these new kinds of segmentations. And this is what I think, I just encourage you guys all to figure out how you can look at the images. And you guys know what to look for in these images. How can we pull that out in an automated way? And we can help you do that. Okay, so that's the human sciatic nerve. That was kind of a fun case because really, really hard to segment into this where you have this mixture. Yeah, go ahead.
How big is your training size?
Say it again?
Your training size. Like, because what we're actually dealing with is like actually small area, and then the neuron are huge. So we actually don't have many training data.
Yes.
So how big is
The training data is, the real training data is 0. I'll show you this in a second. So this is key, right? So like I said, there was 0 hand labeling for any of these things to work.
Well, I guess you needed because we are looking for neurons, so you will need like how many like neurons you need to be able to like segment. We only have like maybe less than 10 in one image.
We need 0. That's the
Okay.
beauty. I'll show you how that works in a second. So that's a perfect way to switch over to this. So right, to make deep neural, any kind of machine learning to work, you need training data, right? And so what's cool about our system is you don't need training data that's done with like, here's a picture of my thing, and somebody goes in and hand labels it or hand defines it. That is not what we use. So I'll show you what we use in a second. Okay, so we also had a Xenium mouse brain data set. This is one of the early data sets, again, that Aldrin and through Aldrin and other groups got these going. And our very first approach to looking at this was to ask questions about, can we just pick out, just based on density and based on these striated patterns, can we pick out the regions in an automated way? So this is not automatic region identification yet, which we'll get to. So here is what the very zoomed out view of this looks like. Again, 10x Xenium, and these are the nuclei. So one of the hemispheres of the brain, you can see the cortex on the outside, the hippocampus and the midbrain on the inside. And so just looking at this, you guys already, those of you who've looked at brains, kind of see the structure that's there. And here's what this looks like zoomed in. It's kind of like the picture I zoomed in on, showed you earlier. And through, again, this physics-based approach, here's what the classification looks like. It's very similar to the CAN classification and the cell-pose classification. The only difference is it works quite a bit better on the really, really high density regions, because those are quite hard to segment. And these algorithms that we have can handle that because they've been trained on that. And so this is what comes out of it. So here it's just segmenting every nuclei, but it's giving us 2 different classifications of like the lower density nuclei and the higher density nuclei. That's the blue versus the pink that you can see in this image. And so not terribly, terribly useful, but again, top down without knowing anything about transcripts, this just instantly pulls out, and you can see the granule cell zones where all the nuclei of the neurons are living, in this case, mostly in the hippocampus, but a little bit also in the cortex, in one of the layers of the cortex. And so, of course, we had this, and I said to Lena, tell me what are the top genes that are differentially expressed in the blue cells, sorry, the blue nuclei versus the red nuclei. And those of you who know something about brain morphology, so we did that just inside the hippocampus, so in a region right here. And all of the neuronal nuclei live in the red regions, and it's the glia nuclei that live in the blue regions. And so what we get out as the top hits is an oligodendrocyte marker, an astrocyte marker, and another like generic glia marker are like the top differentially expressed genes, right? So this is something that we did in really quickly by applying the segmentation and asking questions about these 2 different classes. Probably not 2 classes that are necessarily useful to you guys, but just as a quick example. And again, it's only top down. Yes?
Yeah. I'm wondering what is the intensity threshold or cutoff you use for differentiate between the background signal and the intensity?
So you said you're asking about the threshold?
Yeah, QC, quality control to distinguish between the background signal and the real intensity. Do you use anything like
No, because I'm just doing this on the image, right? This is only on the image data, and then asking questions about the transcript. So the thresholding is just on the image, and since this is this is called instance segmentation, there's no like built-in number, right? This is not an algorithmic-based segmentation. This is a deep learning-based segmentation. I'm not sure if that's answering your question.
we consider all the intensities likewise? We do not want to cut off anything?
Correct. Nothing is cut off. We're seeing the full image. Yeah. And that's what makes it good at dealing with these large variations in intensity that we were seeing inside of the sciatic nerve. Right? So it's because there's not a single threshold that we're using that would normally throw out stuff and keep other things. Right? So this neural network is adapting to the fact that like intensities can be different. In fact, in the training data set, the intensities are all over the place. So it understands that you can see things even when the background is really high or when the background's really low. Yes?
Is that normalized to like one z-plane? Like how do you handle the depth?
Yeah, so we can either do it at a single z-plane or with a max intensity projection. So for most of these tissue, it's such a thin slice anyway that you get a similar kind of result. But yeah, we can either do it on the max intensity or on a z-stacks, basically. Yeah. Great, you guys are thinking about this. Okay, so here we actually have data and it works nicely. The new thing that we're working on is, those of you who do this with brains, you have to draw the regions that you're interested in, right? I want to look at this area of cortex. Somebody's got to say, yeah, here's that part of the cortex and compare it to others. If you have a neuroanatomist that you work with, that's fine, but it still takes a lot of time, and you're still usually only focused on a few areas because it's just very time-consuming to go in and draw these. If we could automate this and bring you guys all of the regions, that would be wonderful. There is a few papers out there that do it, like Deep Slice, but they're really tricky, and there's a lot of issues because of like sections are at slight angles. And so here's where I think I'll do a better job of addressing Guyon's. So here's what, in Allen Brain Atlas, here's what one of these reference images look like, and you can see, even in this one coronal slice, hundreds of different regions that a neuroanatomist might want to identify or look at. And so what Lena did is actually to go through and download all of that data and use it to generate basically all the polygons or all the masks that define these different nuclear regions, right? So this is something that we all have data for. And this goes to Guyon's question. So for all of these things I've told you about, no one is hand labeling and no one is teaching it by hand what to do. Instead, we have simulation engines, and those are what build the architecture of whatever it is we're interested in, right? So whether it's the architecture of a brain, of a sciatic nerve, it's usually very, very abstract things, like just clusters of cells, or stria of cells, or bright cells and dim cells, usually, But those are then, we apply optics and physics on those images to turn it into a microscope-like image. What's great about 10X, right, there's certain, like any platform, there's certain like little tiny things that you can notice, and so we can apply those same transforms, so we get images that are very much like a 10X image. And then the neural network learns to back that out and get you back to the ground truth. And since these images were made, right, nobody had to like hand do them. They were designed from scratch. They were simulated from scratch. We have the perfect ground truth, right? So the annotations are at an infinite resolution because everything about them was defined from us. And so the answer for like how big the training set is, it's insane because we can generate millions and millions of training images, at the snap of a finger, once we alter the simulation so that it fits whatever framework we're working in. And so this is just a little view of what that simulation, how we generate the simulation, what it looks like, and so which of these is the real brain and which of them is the simulated brain. You can see that like things are tilted at slightly different angles. When you go all the way down to the flashing of color here is because Each of these is different simulations. The neurons are all in different places. They all have different subcellular properties. The tilt, the skew, the different regions of the slices, right? So this is how these simulations work, right? So we can basically rebuild a 10x xenium image of a brain at any slice, at any angle, with any piece. And now the neural network is going to understand the architecture, basically, now from ground up with just, and if it doesn't work, we just add more training images and it gets better and better. So that's what's really cool about these physics-based techniques. Yes?
Sir, is this based on mouse or brain?
This particular one is based on mouse.
Yeah, because what if you don't have a brain image that doesn't have a perfect structure? Is that going to work for that too?
Do you mean like part of the section is missing or is split? Yeah, so we haven't built that into our simulations yet, but since we see that in all the slices we've used, that's something that we can build in, is like tears in the sections. A lot of different fixation techniques cause like little rips in different pieces. So that's, again, we can add those really easily into the simulations, and now the neural network can understand that, oh yeah, here's what it looked like before, but now here's what it should look like after corrected. And so that's kind of what this cool picture is on the top. So anyway, I'm out of time. We've done this also in spinal cord, where we can identify regions of spinal cord. We're doing this even in mass spec imaging with intestines. And I'll close with these are things that we've done now in several different places for, not in tissue space, but it's been really exciting to move these physics-based techniques into the arena of spatial, and just like so many questions that we can answer in so many ways to look at the biology. So I am excited for us and you guys as you try to, just like Rich said, like this data set that you guys are going to generate when you bring a tissue, it takes time, right? You've got to work with people on the front end to get the tissue just right. You've got to work with GTAC to make sure that everything there is done just right. And if you want, you can work with us and others to analyze this data. But you guys, there's just so many questions we'll be able to ask in these data sets. I'm just really excited about it. So thank you.